eat it up and clean it up, but the cement kind of stays behind. No one eats that.
So is that a protein secretion? Is that?
I don't know.
Yeah, this is something they make themselves, right? They don't go to Home Depot and buy a little container of super glue and.
Wouldn't it be like
It's
two-part
right
epoxy?
next to the rubber cement is octopus cement.
Right, right.
Yeah.
And the shimmer that we're seeing is warm water that's associated with the areas that the octopus are in.
I just took numbers.
Sort of like seeing the shimmers on a road on a hot day. And so if that water is moving around there, then the octopus don't want those eggs to be getting blown into the current. So use that cement.
Nice.
The glitter you see is particles in the water being hit by the lasers on Hercules. And they sparkle when they go through that laser beam.
All right, video, go ahead and come line. I'll look down this feature here.
We're seeing some black corals there, yeah?
And there's a sponge, Colophecus.
There's this.
Oh, there's Amanda.
Is that the one with the red center?
It's actually a little bit blocked by the porch right now. It's going to be just to the left of that bottommost black coral.
Was that a Z1 sponge?
Yes, it is. It's a big white sponge on a white stalk.
Sure.
Somebody wrote in
there
all
it is.
caps, who is the smart lady telling us what everything is?
Yeah.
The amazing Amanda.
Yeah, that's right.
Oh, I
Alright,
only
video,
talk
go ahead
about
and
the
push on
sponges, but we've got Erica here. She's an expert in corals and the fishes here.
Video, go ahead
I
and
think
push in,
everybody's
please.
got their specialties on the ship. It's a team effort.
Look at that great
Look at that
sponge.
sponge,
that
Amanda.
sponge.
Wow. Oh, cool.
That's a nice looking osculum. Yes, it looks like something straight out of a Dr. Seuss drawing.
Yeah, absolutely.
Yeah.
Or something from outer space.
You guys want to see inside of it?
Sure.
See if there's a
Sometimes
shrimp home.
animals will be inside of sponges, other animals.
Too close to the bottom.
A lot of people oftentimes think that that large hole is where water is going in, but
Right.
that's not the case, is it, Amanda?
No, it's actually the opposite. So the water goes in through all the other parts of the body wall of the sponge and passes through canals and filters within the body and then comes out of the body into that inner chamber and out through that hole, which is called the osculum.
And I remember you telling me that they filter the water at an amazing rate, and the amount of bacteria that is removed is remarkable.
Did we get a shot with the lasers?
Yeah, so sponges can filter about 900 times their own body volume every day. And from that water,
Can we
they
zoom
can
in on
remove
that male?
95% of bacteria from it. So they're really changing
Sorry, on that
water
octopus.
chemistry
Just
around
check
them.
if it's a male.
Oh, I was curious.
Yeah, go ahead, video. That's good.
Hope those little third graders are listening and are soaking it all in
Yeah.
like little sponges.
Close that little arm. So we're looking at this octopus here. Try to see that arm that's curling up behind on the left side to see if it's modified, and it looks to be modified.
It does.
It doesn't look like it has
So
suckers
this
on the
is a
tip.
male octopus.
I think I saw a hectocautalus.
Yes.
Did any of you?
Yeah?
Yeah, definitely.
All right, guys, I'm going to start
Yep.
going on the
Okay,
transect.
sounds
Looks
good.
like we got a good move on. Go ahead and come wide, please.
Thank you.
Is that Mike?
Probably got to turn Argus around right here, so I
Wow.
think it just slopes off.
Yeah.
You're not going to be able to fly out ahead at all.
Yeah.
And I have to turn around the long way, which I think is going to mess you up. See what it does. You
Sure.
want to have a negative half turn,
Yeah.
or be on that side anyway.
Looks like in the distance we can see some more octopus. Do you see the?
Mm-hmm.
Yeah, it's nice how they
Yep.
illuminate. Or glow, I guess.
I'm actually going to go the other way. Stand by, we're just approaching. I'm going to approach
Yeah,
the
we're just
wall
flipping
again.
the vehicles around here.
Okay.
So
Wow, I'm all tangled up.
Oh, look at that nice crack.
Yeah.
Wow.
If we could,
Wait, is it a crack?
that looks
It's
like
just
a,
like
Might have
where the
just
rock
been
comes out
sediment.
of that sediment.
you
Yeah.
want a sediment core here, or is this not what you're looking for?
Is this? I don't think this is be thick enough for you, Dane.
Um,
I'm thinking it's probably just a thin layer.
yeah, I think it is. I think farther off the flanks is going to be better.
All right. So long
Sanctuary.
hair,
Driving ahead.
on
Sanctuary.
the right, we're seeing some arms, octopus with their arms out, and then we see others that have their upside down brooding posture. So kind of
Sorry?
a mix.
Yeah. I'm just trying to get ahead here a little bit. Spend too much time on that octopus, I think.
Well, if
Shout out
you-
to the
hold on,
sanctuary
Jess, if you're-
folks
if
from
you're gonna-
Lauren Parker.
Was Lauren Parker a school?
Oh, hi, Lauren.
I think it's a person.
Oh, Lauren.
Yeah.
Lauren
Oh,
was with
Lauren.
us last year.
Hi, Lauren. Took me a moment. We've been hearing a lot of schools.
Lauren was at UCSB too. Great to hear from all the people out there.
I think thought I saw another one of those ctenophores, but I'm not sure. So are we pointing the direction of our heading right now?
Yes, we're currently pointing the direction, and we're just discussing which way we want to do the vehicles up. So
yeah,
we're going to
okay.
have Argus swing it over Herc, go into a bit deeper water, and then we'll flip around and go backwards down.
Okay.
So that means we have a brief moment here for us to look and see what you guys want to see. Sure. It's off currently.
In this shot,
And
you
back on.
see in the
Thank you.
upper portion, you see a cuskiel, that kind of ghostly looking face and kind of deep body. And then to the lower right, you see a different kind of fish. I'm thinking that's a grenadier. Ooh, and then a beautiful sponge just came into view.
You guys want to see any one of those in particular?
I think the sponge.
Sponge, sorry fish.
Yeah.
Go ahead and push on in a bit there, video. The fish really wants to be seen. I'm sorry, guys.
Yeah.
Amanda, what are we looking at?
Then that's a twofer. That's perfect.
I don't know for sure. My best guess is heteroreet, which is a glass sponge.
Thank you.
That is a beautiful shot of the cuskiel too.
Yeah.
And a couple octopus tucked in, you
I'm
see?
gonna bring the tether actually a bit tighter, Jess. You're
Yeah.
right under me.
Sounds good.
Can we pan down just a little bit if you have a chance? There was something on the seafloor that caught my eye.
Sure.
Or I guess we could zoom out. So that I
Yeah,
can
go ahead and
take
come
a
a little
look.
wider, video.
That, see that just kind of blob? It's
Potato.
kind of
Oh,
grayish,
yeah.
grayish purple.
Sure. Go ahead and zoom in.
Some kind of flat coral?
It's a sponge.
It's a sponge. All right.
It's pretty impressive that all around it it's covered in dirt, but the sponge is able to keep its body surface clear.
Yeah, right. That's amazing.
Does it do that by pumping out the water that it filters in?
Sorry, by what?
You said it filters water, so does it stay clear because it is expelling water that keeps the sediment from settling?
Possibly partially, but it doesn't expel water across all of its body surface. So I don't know for this species, but for some, they secrete mucus on the outer surface, and the mucus will gather up sediment and kind
You
of
can go a
roll
little
off the
tighter.
sponge.
It's a great shot of those beautiful tube worms. Too.
Yeah. Serpulids.
Sorry.
What kind of tube?
If any students from my invertebrate zoology
serpulid?
class are
I think
watching,
they're
we learned
serpulids.
serpulids.
Yep.
That's them.
Come a little wide, video. Bouncing around too much. Get you guys all motion sick.
Seamus Morrison and others are asking, what are those palm frond looking things that we're seeing?
Go ahead and
believe
push on
that-
in there, video.
The larger ones that we've been seeing have been black corals.
Deep sea corals known as black corals.
Go ahead and go in a bit tighter.
It's a
Someone else wants to know, why are the lasers green?
The green light travels far underwater. So there are red lasers as well. Some vehicles have red lasers, but you can't really see out as far with them.
That's a big sponge.
Yeah, because those lasers are 10 centimeters apart. So if you can see what a small portion of the sponge is 10 centimeters, then you can use that 10
Yeah.
centimeters
That
to get
gaping.
an overall size.
10 centimeters is about 4 inches, so
All right, go and come a little wide video.
that's a big sponge.
Look
Yeah,
at this
sure
black
is.
coral over here.
Amanda, how would you describe the morphology of the sponge?
Well, down here we were actually just saying it looks like a cauliflower, so I guess
Okay.
cauliflower sponge is as good a name as any.
Okay, sounds good. Thank you.
It does
Yeah,
make me
okay.
hungry for cauliflower, you're right.
Go ahead and push on in their video.
And no, Timmy, its pants aren't square.
It's good.
Oh,
So
that Timmy.
this is a black
Black
coral,
coral.
and if you look closely underneath all the fleshy polyps, you see that black skeleton underneath.
Are collections possible right now? Like, is the ship
We're waiting for
ROV
the ship
stationary enough?
Move.
It depends on what you want to collect. Do you guys want to take a piece of that coral?
Actually, a small piece of the sponge.
The large sponge.
Small.
We can try to do a really quick grab.
Okay.
I think you're gonna have to stop the ship. I think you're gonna have to bring it back.
No, let's not do that. That's a long
It
process.
is a lot of time, yeah.
Okay.
Yeah, there's
Alright.
everything's moving now. So
Okay.
that whole like 10 minutes we've been here, that's kind of how long it takes to get everything going at this depth.
Okay.
Okay, that's
Sorry,
good to know for
Amanda.
next time.
So now we're going down our line backwards.
Yeah, because
correct.
right now it's pretty flat for the next few minutes, but then it just sloughs off, and if we go down frontwards, you won't really see anything.
Cool.
Annie, somebody is saying, "Get that science done!" Exclamation point. Also,
Hi, Trevor.
You betcha.
Squeezer's good? Question mark.
Oh.
Yeah,
Oh.
5 of the 6 triggered. So we're pretty happy.
So we have 2 from one location and 3 from another location.
We have 2 from one location, 2 from one location, and then 1 that triggered ambient water,
Oh, okay.
which is
Got it.
nice because then we have that
Like
sample.
drive around just
Great.
or
Is that like, would that be a control?
Yeah.
sniff around a little
Alright.
bit.
It was close. It was close to a seep area, I think. But off, is that right?
Yeah, so the
Yeah.
first one that was taken at the vertical vent, and that one was in fluid that was about
Can we try to zoom out just a
9.5
little
degrees
more?
C.
Sorry?
Can we zoom out a little more and just see if we can still see well, but get a little more area? Here's
Sure.
a shot.
Yeah, video going full wide. Oh, it's right there. I think so.
And then the second one was in an area that was about 6.6 degrees C, so a little cooler. But that was apparently in the octopus spa. They
Right.
had 2 good fires on that one.
Right. Yep.
Other door. There's some broken stuff over here. There's something that looks broken over here.
Oh,
Egg casings or maybe shells.
Where are you looking?
Right
Pretty
there.
much.
Yeah. Like
Yeah,
teeth
down
laying on
here.
the bottom.
Oh, uh-huh.
Go video and zoom on in. Let me come down a little bit.
Maybe serpulid shells.
Maybe it's a broken squat lobster?
Yeah, I was thinking
That too.
Okay.
And what is that frilly thing that looks like a
Is that an acorn?
That's an
Dutch
acorn worm.
iris?
Acorn
Yeah.
worm for that guy.
Yeah, that might be parts of maybe a crustacean, huh?
Looks like it. Okay, go ahead and come wide video.
Guess he didn't taste very good.
Is that a sponge, the blue flossy looking thing that was just up?
Oh yeah, go ahead and zoom on back in real quick.
Yes, it is.
Blue flossy.
Down at the bottom of that
Yeah.
shot. Yeah,
Mhm.
that's sponge as well.
Go ahead and push on in a bit more. Yeah, I done it.
Cool.
Oh, and there's a worm above it.
Yeah, there's a polychaete swimming.
Yeah.
All right, go ahead and come wide.
Let's keep going.
All right, I'll start bugging downhill now.
Okay.
I think we've already started.
Yeah. If it's possible, I think we can be a little further out away from the rock and still see the octopus and maybe get a little bit wider view.
Do you mean go a little high off the ground?
Yeah.
Sure. Okay, I'll maintain this heading.
There we go.
That's great.
Is this good
Yeah.
for you guys?
Yeah, that looks good.
Okay. Do you want to just push in around the artifact, around the, yeah, little wider, just until you're clear of the artifact. Yeah, that one on the lower left. All right, I'm starting to maintain a constant heading for your video transect.
Okay.
There's some whelks and a
Yeah.
little octopus in that
Right.
area.
So what we're doing now is we've started a transect that we've already determined before we came out here, and we have a series of them that are covering this larger feature of rock. And we're basically looking to get a sense of how extensively the octopus are distributed across this rock, and if there are other areas of high concentrations of them, and to get a sense of the other organisms that are associated with the octopus, especially when they're in these denser aggregations.
We're seeing them pretty consistently.
All you have to do is play this in reverse and you get a real transect.
Guys, we have a problem here. Somebody's crying out for help. I'm getting addicted to science and can't get any work done.
Oh, I know. That happens to me.
And you have to become
Well,
a scientist.
Yeah, the only solution then is to get more of your drug.
Join the club.
There you go.
Join the club of exploration and science and discovery. It's fun.
Is an OET accepting applications now for their various fellowship and internship programs?
They are, yeah, there are a lot of different internship programs.
Yeah, I was just trying to clear that ledge, but I can start working. Yeah, I can start working up. That's a good point.
Just so exciting to have all of these listeners from all over the world joining us. We've got a few folks from Reykjavik, Iceland,
Oh,
Annie Hardy and Arlene.
Oh, hi, Mom and Dad.
Huh?
It's my mom and dad.
Really?
Yeah, they're visiting Iceland.
Oh. Well, hello.
Awesome.
How nice they're taking out vacation time to give a shout out.
Yeah.
That's great.
Yeah, they're big support. Appreciate their support a lot.
Aw.
Did we change direction or heading or something? It just felt like that we moved a little differently.
Negative. All we were doing was just biasing up slope a little bit
Okay.
so that Argus doesn't
Yeah,
go
watch,
into the
maybe
cliff
have
side.
high pack up, Jess. You can use that to.
Yeah, I see on high pack.
Yeah.
I just wanted to, I'd rather bias towards the slope than away from the slope.
Okay.
Yeah, this is fine.
Yeah, I think Argus is like pretty much right on that line.
Yeah.
What's that?
Ooh, they got orange, like yellow.
Yellow. We haven't seen that much before.
Amanda, if you're still listening, somebody wanted to know the age of that sponge.
Oh, goodness.
Oh, you guys ask all the hard questions. I don't know the age of that sponge. What I can say is we know that in general, glass sponges grow at rates between 1 and 3 centimeters per year. So pretty old, older than me for sure.
Here we go, got a little math problem.
Curious what that stalked yellow thing was. Anyone know?
I have a question for the Mumbari science group. Have a lot of the identifications of the organisms been from sample collection and then like laboratory investigation, or has it been a lot of just observationals?
Yeah, it's been
Identification.
through collections, and then they get sent out to various taxonomic experts for certain groups. And once their identity is verified, we have a big database, the VARS database, which are the photo references. So then we can cross-reference all the photos in that database and get records of that species and be able to apply the right name to it.
So the organisms that we've been seeing on this dive, are they what you expect based on that database?
Yeah, sounds like it from previous trips out to Davidson Seamount. These are the species we'd expect.
A lot of what we're seeing is also in a guide that we've put together for, well, that Erica has been the lead on putting together for all past observations at Davidson Seamount. But Erica, I was trying to find that tall, stalked yellow organism.
Sponge?
No,
Oh.
I don't think that was a sponge. I thought it was a cnidarian of sorts. Don't know if she saw it, but I was curious to try and find it.
It was kind of forked looking.
Yeah, the yellow.
Yeah. Mm-hmm.
It's not the Bathypathes, right? You mean a different one?
Yeah, it wasn't a black coral. We just saw it briefly. We didn't zoom in on it.
We're not sure.
Okay.
Which one you meant.
Say goodbye, Annie.
Bye, Mom, Dad. Love you.
Mike, what kind of
Yeah.
technology do you wish someone would invent, either for sample collection or exploration, etc.?
Got a few octopus down
Wow.
there to the below this ledge.
Yes. Is that enough on our transect line? What was it?
So see to the right of the ledge, we've got a little
Second,
looping
sorry.
down there. It's possible to just peek around to see if there's more tucked down in there. Jess, are you talking to us? We can't hear you.
Sorry.
Okay. Sorry.
Do you guys, well, the question's kind of irrelevant now. Never mind.
Okay.
I just wanted to peek. I don't want to stop what we're doing. Just get a little bit of a wider view there to
Sure
see
thing.
if it was worth. If it had been a lot more there, we might have wanted
down.
to.
Yeah.
Look how
investigate.
right at the edge of that drop-off, right below the top flat part, there's a lot of stuff living down there on that vertical edge where it doesn't get covered with detritus.
Hello, Jenny.
If you guys want any of this marked as potential features of interest, let me know.
Okay, we'll do.
Maybe that's scarp.
Uh-huh. But it is not an area of high density? What do you think? Oh. Yeah, go ahead.
Aaron, you might just want to make a marker that this is a place that has a pretty significant drop-off region where we noticed maybe 10 octopus clustered together.
Okay.
Thank you.
Yep, no problem.
So,
It looks like it's protecting it. It looks like it's,
So?
yep.
When we see octopus that are out and moving around, we're just taking a look at their arms and how they're using them and trying to determine might be a male or a female because they have an anatomical difference that we can see sometimes.
question is there only going to be one dive in this area?
This is our first dive of this leg of the expedition, which is the last leg of the expedition. This one's gonna.
And is that you? Can you
Oh, am I
a little louder
quiet?
there?
Can you hear me now?
A little bit louder, please.
It's right next to my mouth.
There we go.
Okay, is that better?
Yeah, much better.
Okay, so this is the first of 2 dives that we're planning out around the edge of Davidson Seamount. The first one we're on right now. We're going to be doing these transects now for many hours to take a look at the area surrounding the octopus garden and some other features that we've identified where there are high concentrations of octopus. And next we're going to do a second dive that will not be on this feature, but it kind of starting at the north end of it and traveling around the base of Davidson Seamount to look for other interesting areas that may have similarities to this site or maybe totally different. We have to go look to find out. So those are the 2 dives we're planning for our time out here on the Nautilus in Monterey Bay National Marine Sanctuary.
And this line is going to take us kind of down the slope and into what we suspect is probably more sediment. Right
This
at the end of the transit.
Definitely some of the other lines will. Let me see about where this line terminates. Might be,
Oh,
but
yeah.
we may need to get into some of the other transect lines to get to thicker sediment areas.
Seems like it ends just as the slope is starting to peter out, but not so much as being a flat area.
Okay.
Aaron, the transit line that we're on, that's the red line that we're seeing on the HIPAC
Yeah,
survey screen.
It's the red line, and the ship is offset from it because Hercules is dead on it.
Okay.
Can we peek a little to the right?
Sure. It's easy.
I see some sea stars and some octopus caught
Yep.
my eye. Get more of these rubble-y areas is sometimes where we found them in larger aggregations, but it looks like there's just 2 right there. Okay,
Good.
thank
You
you.
know,
What?
Yeah, that is a drop off.
So if you're just joining us, we're at the Davidson Seamount in the Monterey Bay National Marine Sanctuary, about 50 miles off the coast of Monterey. We're currently running a transect of the seamount. Earlier we deployed a number of different pieces of scientific equipment to sample the environment. Some we put out and retrieved, and others we left, and they will be retrieved in about a year when we return. Somebody wanted to know where the, who came up with the idea of using pink flamingos to mark sites. If you were with us yesterday,
No.
Oh.
they were talking about using pink flamingos to the
I can see if it's consistent.
Mbari.
It might also just be the vehicle.
Wait, was Mbari that used pink flamingos?
Negative.
I may have missed that conversation. I don't know the
Might just
history
be a
of
feature too.
deep sea
so.
pink flamingos, and science.
If it's an Argus, then
Yeah, it's an Mbari thing.
It's
Got
an
the little
Mbari?
Mbari logo on the
What are you
side
looking
too.
Oh, okay. Can you say that again,
There was just
Amanda?
a false return on Herc, or maybe it was real, but it might have just been the small rock. Or it could potentially be you. Depends on our
Definitely it was
delta.
somebody from a suburban environment.
Especially because that sonar is on top of Herc. Yeah.
They were leaving for a cruise. They forgot the markers, and a home improvement center was the only thing that was open with lawn decorations.
And how did they know that they were going to be negatively buoyant?
But these ones are interesting
that?
because they're hanging out upright,
Mm-hmm.
just around.
Sure.
Ooh, is that a wall to our left?
Yeah.
What is that globular thing?
Yeah, do you guys want me to change my heading and look?
Yeah,
Yes,
please.
please.
Oh. We're going up against an escarpment.
There's one little octopus in a hole over there.
Yeah.
It seems to have gotten pretty sparse there.
Mm-hmm.
All right, coming back to transect heading.
Just about as many upside down as right side up
Yeah,
in
you
this
know,
area.
at some point it might be worth, if we do find one that's brooding in a hole or a couple, just zooming in and see if they have eggs, see if we see any shimmering.
Mm-hmm.
Okay. Sounds good.
Sure.
But that might be, I mean, when we get to the end of the transit line, because we're going to be change things up anyways.
Mm-hmm.
Yeah, sure thing. Sometimes it might be easier at the transit line, because at the end of the line, rather, just because we are moving at a constant speed.
Yeah, like if we see something in the screen and we had asked you to stop, that's not like that's a delay, right, for you guys?
We can have, we can have a couple of minutes, like if we have a good position, if we haven't already been stopping and looking at a few things, we have a couple of minutes, like window where we can stop and then catch back up with the ship, but it's pretty limited.
Okay.
But pretty soon, because we have a lot of layback right now on the cable.
What does that mean? You have a lot of layback?
Just means we have a lot of cable out. Yeah, because I think Argus will now swing for a while to get under the ship again.
Yeah.
So you're saying we'll be moving slower during that
No,
time, or
just
no?
when we get to the
Okay.
end of the line,
Oh.
it'll take a minute or it'll take 5 minutes to let us swing everything out and
Okay.
move over to the next line.
Okay.
It's not that slow going once you're on. Like you can see, like once we got this one started, it goes at a pace, but it is a fair bit of time to set up a line and get started on it.
What is that thing to the left?
The dark.
It's very
left.
dark.
thing,
Oh,
yeah.
maybe it's
Oh,
a
sea
cucumber.
Sea
I think.
cucumber, yeah.
That's a
Some of these black corals are pretty good size.
ooh.
What's,
What
yeah.
is that?
Was
Oh.
it Cuskeel maybe?
Or
Oh yeah,
yeah,
just with
or
the light.
grenadier maybe.
Oh yeah.
Sometimes the shadows can be
Make
deceiving.
it, yeah, like I thought it was.
Kind of looks
it-
like a Dementor.
Science, just thinking ahead a little bit. Do you want to move, which transect would you like to go to next?
Right. Just thinking about that. I'm kind of leaning towards going down to, which one is that? Well, but the octopus spa is up there. So maybe we should go, you want to go
Look,
down?
right on the ridge.
Annie wants to go
How's
down?
that?
You're right
Okay.
on the ridge, sonar.
So
See
we
this
can
thing?
go 8,
Sounds like
10. We can go to the one that
Yeah.
goes between 9 and 10. So to point 10. Well, my 10 would be your 5, I think.
Sure, yeah, so the one that's to the south.
Yeah.
Yep, okay.
For Dane, may or may not. It may. That may get us to where we can take, we might get enough
Okay.
sediment to get a sample, which would be
Okay.
good to do that sooner rather than later
Yep.
too.
So
Yeah.
then are we going to be on the north side or the south side of the seamount?
Of this feature?
Yeah, or the east or the west, I guess.
So if this is the feature we're on right here,
Yeah.
we're going to move down here and be moving along this
Okay,
next
so
line
yeah,
down.
on the southwest
So
side.
it'll be
or
this.
Aaron, is
southeast.
the ship stopped now?
Southeast. Yeah,
Ship
Yeah.
is stopped.
Yeah.
There's
Yeah.
this little
this is
sediment
fine.
pocket
And let
here
that
that I'm
swing out a
looking
bit
at.
and then
Okay.
go down to the next line.
Okay.
So we are doing a portion of 1/3 of 7/8 so far. And then we're going to go on to 9/10.
Are you guys considering looking for a sampling location at the end of this line? Did you want to hang out there, or you want to move to the
Well,
when we're ready?
only if we see, like, right now, do we have time to zoom in on that brooder one right there?
The one that's coming below center screen here?
Yeah.
Sure.
So
Standby video.
science team, could you give us a sense of the size of the octopus garden relative to the area of the transect that we are trying to do right now, just for scale?
That's good. I don't see any shimmering, and she's not moving
We're
much. So
occupied at the moment.
yeah.
Yes.
Hold on 1 second.
But that empty hole below it looks
Uh-huh.
like it had been occupied.
Yeah, it looks like there's some residue.
And-
All right, guys, I gotta come wide.
Yep, okay.
Okay.
Yeah, so.
Yeah, you really want to be
We
like
can't hear you.
closer. Move
Yeah.
the camera down.
This
and
is an
get, like,
area that we have evidence
so
that
that-
there's been
Yeah.
an
a
egg
minute or
clutch
two.
laid.
You're right on
Another-
the edge there.
Seeing
Another
the
30
associated
seconds.
shimmering discharge.
Yeah.
Right.
And our viewers may notice it's easier to see the brooding octopus because the white underbody is very visible. Like in the very upper left, you kind of can see that white circular, that's a brooding octopus. But then when we see octopus that are not in that brooding posture, they're not quite as easy to pick up. You see the more as purplish gray, such as the one that's kind of to the middle right of your screen. So we're trying to notice all of them.
Are those sea stars eating a sea cucumber?
Where?
Up in the kind of the center to the left of the lasers.
Can we zoom in a little to the center, to the above and the left of the lasers?
Yeah, do you want to do a quick zoom there video?
Yeah, just quick zoom. A little bit up and to the left.
Sure.
I think it's way over there. Clump of something.
There.
Yeah, that.
Yeah, sea stars.
Snack?
All right, I gotta go.
Yeah,
Sorry, guys.
that's
Come
okay.
wide.
That was more than enough. Just curious what it was.
I wonder if the sea stars are the main scavengers on the site.
Well, and the cucumbers.
It's a nice sponge.
Do they scavenge?
Well,
I thought they're more deposit feeders.
Yeah, but I think they take up everything that's on the surface of the sediment.
But we've also got some crustaceans. We do see squat lobster. The shrimp are harder to see from this distance, but they do seem to be in amongst some of these rubble patches. And the sea stars and the snails that we've
The
been seeing, the
whelks?
whelks. So there's quite a variety of animals here that can help basically, once things die, to consume them and
Recycle them.
recycle them into the food web down here.
The single-stalked organisms, are those corals or sea pens?
They're corals.
They're
That's
corals.
the black coral.
The black
yeah.
coral. Oh,
Yeah.
the long single stalk.
Well, with all the fringes off of it.
Yeah, but there's
No,
a
no,
few
no.
that are just like a single
Yes,
long.
Right. That's
that
I don't know.
is, I think it's in the guide. I know which one you mean. They're
Yeah.
reminiscent of a sea pen.
Yes.
And it's,
Are they just bamboo whips or?
There we go.
Bamboo corals?
I think it's this lepidisis.
Oh yeah, yes, we saw a lot of those last time. Thank you.
You're welcome. So there's a grenadier. You see it has a lot of grayish white around the head, and then the tail's a little bit darker. I think that's the abyssal grenadier. Or is that the ghostly grenadier?
Aaron, you can probably put that move in to go down to number 5.
I just want to confirm you guys don't want to do any sampling or anything at the end of this transect in this flat area.
I don't think the sediment's deep enough
Right.
here.
No, it's not.
Yeah, there's that yellow
Yes.
guy you were talking
Yeah,
about.
Right.
yeah.
So it looks
That's
to
a
me.
stalked crinoid.
Okay, thank you, Amanda. I was thinking
Ah.
What was that, Amanda?
stalked crinoid.
Stalked. That's a
Okay.
crinoid.
So that
go.
those are also referred to as feather stars,
Yes.
right?
Right.
Crinoids.
Mhm.
Yeah, we'll prefer to have Vargas in a little bit deeper water.
I'm
They're
hoping
10
we come
meters
across
lateral.
another
Okay.
one of these aggregations of octopus surrounded by sea stars.
128,
and
Roger.
Collect a few of the sea stars.
So we've
I
reached
have that too.
we've reached point 8.
Okay.
Is it?
All right. And transect point 8. Okay.
It's gonna take a while to catch up, and then it'll take a while to slow down. Might as well just step it, I think.
I think we
All
have
right,
some
Mike,
more
I
of
got
those.
another question for you
Sonar
from the
force.
audience out there. Do you pilots have a simulator to stay in practice on the ROV and ARM during the off season?
No, we don't. Some oil and gas operations do that, where they have so many systems and employees and stuff. But that's not generally the case. I've never really used one in my career.
Yeah, and then we'll
I've
just
used one once or
we'll
twice,
just
but not, you know.
kind of add them on so that we keep the
Practice.
momentum going.
Thank you.
Are we moving towards point 5?
That's correct, yes.
Okay.
There's another one. That's a very good example of
Oh,
the single-stalked
Lepidopsis.
deep sea coral. that we were
That's
talking about.
lepidisis.
Lepidisis.
Yeah.
Yes, very
Go ahead and zoom one
cool.
in here.
Yeah, let's zoom in. And you can see along
Sorry.
that stalk there are polyps.
Once we get it back into view.
And is this a colonial organism?
Yes. And I'm thinking I have to get in a little tighter to count, but I think there's going to be 8. Each polyp's going to have 8 tentacles. 1, 2, 3, 4. Is that
It
an
should-
Yeah, 8.
octocoral?
Mm-hmm.
I didn't see it.
Come a little wide video.
I got some good images of that one.
You see that very symmetrical sort of round? Is that a sponge down there, or? It looks like a
Symmetrical.
golf
Oh, wow,
ball.
look at
Right
that
there.
red shrimp.
Oh,
Hold
yeah.
on.
Yeah.
Can we zoom in on that, please?
Sure.
We had a request for a red shrimp.
There you go.
To
Come a
collect
little
one?
wide video, please.
Yeah.
Yeah, I don't know if we're going to have time to collect
Yeah.
one, but we should get some really good
Yeah, if you
video of it, please.
Sure. Let me just get us so I was a little bit better situated here. All right, go ahead, video.
And are those its legs? Yes, look at
Yes.
that.
Oh wow, there's a little
What?
anemone on top of the
Sorry.
Huh?
opposite outside.
Oh, look at that's a
wide video.
tiny stalked crinoid down there. I have to start small to get big.
Hey, I got a few images of that shrimp with the lasers.
Okay.
So we can get a size on it.
Sorry, I can get you guys a better
Steve,
shot
I did see
for
that circle thing you were talking about. I'm not sure what that is. It's very, very round
Could it be trash?
there.
Oh.
All right,
that.
one more shot there, video. Go ahead.
Okay. I am ready. There we go. Almost has like glow. Are those the
Pretty
eye
well
spots?
emitted.
Yeah,
that
eye
glow?
spots.
Yeah, they're just
yeah.
reflecting light
Yeah.
back.
Yeah.
How are we doing on the stills? You feel like you've got some good ones?
Yeah. I got some with the lasers, and then these are without lasers.
Great.
All right, go ahead and come wide video.
Take the
Let me
bubble
just
camera.
pan
Thank you.
Sure. You want to come full wide, please?
The eyes are reflective like that for some species because of the shape of them, and each little segment of eye is called an ommatidium.
Yeah, it's been doing.
I'm having a little trouble making out everything you said, Amanda, but I think you were saying their eyes were reflecting light like that because of their shape.
Sorry about that. Yeah, that's right. Yeah, so the eyes have different shapes to them, and for ones that are the right shape, they kind of reflect the eyes back. And each little shape structure in that eye is called an ommatidium. So that's why when you look at something like a fly's eye, it looks like it's got many little eyes on it. Each of those is an ommatidium as well.
All right.
The more you know.
You can share that at your Halloween parties this year.
No, I'm thinking Scrabble.
Oh, that would be a winning word.
Ommatidia.
Did you know that they added OK to the Scrabble dictionary?
It's about time.
You mean the 2 letters?
The 2
O
letters.
and K?
It's a valid Scrabble word now.
Gosh.
Well, there weren't enough 2-letter Scrabble words, hey?
There weren't.
Make more.
We got another one of those stocked sponges.
Hello, Caulophacus. We meet again.
Yeah, we're going over to their 5-hour. We're going here.
Wow,
there's
that's
an
a
Can we zoom in on
spread
that
out,
octopus,
spread out
please?
octopus.
Look at that.
Stand by video.
I think that's a modified arm.
All right, go ahead.
Yeah.
Is that a male?
Maybe.
Yeah.
It'd be really nice to get in closer if we
But
can.
a very typical
Swing
looking
around.
pose for the males, yeah?
Yeah, look at the tip of that. Is that?
It looks curled up. I honestly can't.
Go ahead
Okay.
and push on in a bit more video.
Is it just what we've been
Oh,
seeing, or do
yeah,
they typically
yeah.
tuck it away?
Well,
I don't know. It seems like a lot of them we're trying to find. It's like the one tentacle that.
Right.
Yeah.
yeah.
I feel like they have a lot of good reason to protect it.
Yeah,
because of
but-
its very
Yes, that's
specific
right.
function.
But there it is. It's the only one that's not being used to maybe to interact with the seafloor, but it does look to be modified, so it looks like that's a male.
That's the hectocotylus, or the
A little wide video,
pseudophallus at
please.
the end.
Yeah.
Get better in the frame.
So we've got another male here.
It's a funny looking one.
There we go. Okay, I'm taking some without the laser.
All right. But you're right, often we have trouble seeing the hectocotylus because they do seem to have it curled up or tucked under
Right.
themselves. So it's nice of him
Thank
to
you.
nice of
For
him
the
to get
zoom.
give us that
Yeah.
that easy clear view.
Yeah, that's pretty nice. All right, video, go ahead and come wide, please.
So along the way, you can see we're also seeing anemones, and we're seeing sea stars and sea cucumbers. A lot of these animals are here. They're either feeding on the marine snow that's falling through the water and capturing it from the water, or they're kind of suctioning it off the bottom after it's settled down onto the seafloor. And then some of the animals we're seeing are scavengers that help clean up some of the larger animals after they've died. All of this is really
Will that turn and, like,
recycling.
drive where you're going with
Recycling
this?
things that are raining down from the
That's
surface.
all right, I'm just lateraling along here.
And integrating
if you're
them
going
into
to cover
the
any amount of
deep sea
distance, like face the way you're going.
food web.
Sure.
So, and the octopuses are probably predators, right? And how about the whelks?
Whelks can be predators.
Yeah.
Yeah,
so
definitely.
a few predators and some scavengers, and productivity is coming from elsewhere.
Right, and some of the fish we're seeing likely can do both, be
Right.
predators and scavengers.
Right.
Is the primary productivity in the surface water above Davidson relatively robust? Do you know much about what's going on above, maybe sinking down?
Well, a
Same.
year ago we were out here and did a separate cruise where we were looking at that because seamounts can interrupt the flow of water and cause localized surfacing of deep water called upwelling that can feed the food web and kind of cause more plankton and then basically start the food web, kind of ramp it up over the seamount. So we were looking at that, and I haven't seen all the results of that yet to confirm that that's the case. But we're definitely looking into that. And if you're causing those
Go ahead and push in there,
localized blooms at the surface, you'll also increase
That's good.
the
I'm just going to look at the coral for a minute.
So
raining of the snow to the seafloor.
aerial surveys that the federal government did along the Pacific coast found aggregations of seals and sea lions and seabirds associated with topographic highs on the seafloor. So they are localized areas of high productivity that feed even surface feeding predators and consumers.
Okay, what is that cool spiky thing down there, Amanda?
I was just going to say, we haven't seen many sea cucumbers. I mean, I'm sorry, sea urchins, but we did have there we go.
Is that a deep sea urchin there?
Yeah, we hadn't seen many of them,
That
but
is
there's
incredible.
It's a little one.
Yeah.
Wow, with very long spines.
All right, video, go ahead and zoom on in.
Stay away.
Okay.
Wow.
Cool.
Yep.
There we go.
There we go. I got
I'm
some
looking
pictures.
for it.
All right, I'm
Yeah,
slowing down.
I was surprised that we hadn't seen any sea urchins.
Go ahead and push on
There
in
we
a bit
go.
more.
Okay, I just found it.
Wow, those spines
A
are intense.
Porosidaris milleri. Anyway, would you like to
Thank
film
you,
it?
Erica.
Yeah, I would love to.
And then
All
what
right,
is
video,
it?
go ahead
Little
and come wide,
shiny
please.
ball in front of it? Is that a
Wide,
little
please.
algae or?
Video, come wide,
Oh,
please.
there's two of them.
Yeah.
They look
I
like
saw
little
those last night on the
marbles.
dive too.
I don't know what that is.
Thank you.
You're welcome. It's a great little Mumbari guide for looking at the organisms that we're seeing.
So was that,
Oh.
did Erica put that together?
Yes, I'm sorry. This
That's
is
Yes.
right. Yes,
Okay.
Erica put that
Sanctuary guide.
together. That's right.
Sorry, Erica.
We're working
Yeah.
with Mumbari
Sweet
though,
piece of
for
work.
sure.
It's very helpful.
Monterey Bay National Marine Sanctuary Guide.
There's another of those stocked crinoids.
Yeah.
I did just see
Mhm.
that in
Yeah.
the guide.
1, 2, 3, 4, 5. And those are related to sea stars. And urchins and cucumbers. They're all in the same phylum.
Echinodermata.
Yep.
So
Request-
we'll be able to start this transect going upslope then, as compared to the last one.
Oh,
yes? Yeah, great. And we've got another bamboo coral and then a sponge there in front of us, the white funnel looking shape as a sponge, glass sponge.
So are we on the second transect now?
Not yet. We're
Not yet.
approaching
Okay.
the starting point.
I'm going to hold off here, Mike.
So finish this step, let it settle in.
Oh, there's a swimming.
Yeah,
Oh, in
we might
Argus
be a
view.
little
Octopus.
a little arf.
There's an
a little
octopus.
off.
Oh yeah, if you look in the Argus view, if you're on view, there's
So
that
I'll come
octopus
forward
swimming.
this way. That was the last step there, was it?
It's the last step that's finishing up. We have about 10 meters maybe left in it.
Okay, yeah, let's see how we settled in.
Do we have time to zoom in on that sponge?
Oh, yeah, sure.
I think Amanda's earned her keep, yeah?
Oh, yeah.
We'll be able to get it from this angle, if that's okay with you guys. Go ahead and zoom on in video. A little tight on the tether, though,
There you
so.
go.
Wow.
Okay.
Go ahead and zoom on
Oh,
in some
that's
more.
pretty.
Yeah.
Some more of those giant protists too over
So
the
some of these
right.
Yeah.
kind of ruffly sponges are Phorea. I don't know if this is, I'm looking in the guide right now. Oh, you don't know what it is? Well, then that's exciting.
Bridge nav.
It's beautiful. Like
Yeah.
cone of lace.
So Amanda doesn't know what it is?
That's what she said.
That is so awesome.
Look at that. It's beautiful.
Yeah. Yeah, look at that.
Amanda, would you want us to try to get a sample of this? I don't know if we can, but... Oh, goodness.
Shift stopped just to see where Argus comes out.
That sounds good. Yeah, I mean, we're like 7 meters off of our starting line on the transect, so we probably will have enough time and tether to take a sample of this. Amanda?
Is there any, Eric, is there any way to ask Amanda if she wants a sample?
Did you want to push on any more on there?
Okay, yes, that's a yes to a piece. If we can get a piece quickly, not larger than the palm of a hand.
Okay. You guys mind if we, we're going to slurp it? That'd be easiest for us.
Yeah.
I think that'll be okay.
Sounds good.
Sure, go ahead and come full wide, please.
We've got crunchy, so are you saying it won't be able to slurp?
Aaron,
Okay.
move the ship back 20 meters
Okay.
back the way it came.
It's
Will
probably
do.
firm and crunchy, but it should be able to be slurped.
Sure. What's that?
This is the time to get one before we get going.
So just like when we're mapping, when we're trying to understand an area better, we'll run transects and go back and forth, sort of like mowing the lawn, if you can imagine, surveying each of the transect lines to better quantify the area that we're looking at.
So if this sponge is similar to some that we've collected on past cruises, they have the texture kind of like Pringles chips. They look really soft, but they're can be really like crunchy feeling.
Well, they're silicious, right?
The glass sponges would have a skeleton made of silica.
Jen, you're barely coming through.
Okay. Yeah, glass sponges would have that'd be a structure. The structural components would be made of silica, so that's why they're called glass sponges.
Can they cut you like glass does?
Science, do you know what suction jar we're going to be on?
I think some of them you definitely want to handle them
so
with gloves.
we have 2 through 7 open, so you could probably put it in 2.
Okay.
Yeah. I'm guessing crunchy too, just from my
Can you fill his
experience
pump?
with you in collecting sponges,
Maybe, I
but
think
can't wait to
it's a
find
bit
out
dirty
when you
there,
have it
I'm
in
not sure.
hand.
Better safe than sorry. Number 2.
Did you guys get any good close-ups of it? I know you're time limited.
Yes.
We did, not from this angle, so before we slurp it, we'll get another one from this angle. We
All right, go
were
ahead.
shooting it from kind of the left side earlier.
Okay, as long as we can see the outside and the inside, that's good.
Okay.
So we can get
Yeah, they
some
wanna
stills of
zoom,
the
yeah.
inside.
You're not on SPL?
Yes.
Zoom in
Yes,
video.
please.
Go ahead and zoom video, please.
Does that look good for the inside?
Well,
It
there
does,
we
yes.
go.
Yeah,
Perfect.
okay.
Nice.
Yeah.
Thank you.
Can you iris down a little to get the outside's architecture too?
Oh,
Okay.
oh
Did y'all
yeah.
hear that?
Great shot.
Okay, thank you. That's good.
All right, that's
Alright,
good. We're
come on,
clear to
please.
sample.
Oh,
Full
no,
wide.
Forgot about this.
Oh, hey!
Caught her
So
sleeping.
long, sucker.
Oh,
No, don't come back, don't come.
Oh, get outta
There we
here.
go, there we go. Oh. She'll find a rock.
And then there's still one on the probe. You can see a little arm sticking up.
Yeah.
Oh, we got